So there are different differences. There are differences between the eukaryotic flagella and prokaryotic flagella. In this class, we will cover the prokaryotic flagella. And since we are discussing about bacterial flagella, we need to differentiate what are the major differences between gram-positive bacteria and gram-negative bacteria in terms of flagella. Okay. So this structure, which is given in this particular diagram, is actually a gram-negative flagella. As you can see, there are 4 rings. So you can see from here there are 4 rings. So one ring is called L ring, L4 LPS. Means we know gram-negative bacteria is having outer membrane, which is majorly having lipopolysaccharides, LPS layer. So since this ring is present in this LPS layer, so therefore this ring is called L ring. And there is a P ring, P for peptidoglycan. So because gram-negative bacteria is also having a thin cell wall made up of peptidoglycan, so the ring which is present in this particular cell wall-like structure is called peptidoglycan ring, P ring, or this layer is called peptidoglycan cell wall layer. Then there is a periplasmic space. In fact, this complete space between an outer membrane and a plasma membrane, between these two membranes, the space is called periplasmic space. A thin peptidoglycan cell wall is also present in this space, and the ring is called P ring. Then there are 2 additional rings. So this is called MS ring. Initially, it was called M ring and S ring. It is a combination of 2 rings. Actually, you can see here. This is S ring, and this is M ring. So M ring is present in the plasma membrane, and S ring is supramembranous like structure. So it is present above the membrane, therefore called S ring. But later it was found that both rings are actually merged together to make a common ring. Therefore, nowadays it is called as MS ring. And there are proteins. So this flagella is made up of flagellin protein. So flagellin protein is denoted by this FLI, flagellin protein. So there are different uh, abbreviations have been used here. For example, F, G, M, N, and so on. Okay, so these flagella is actually made up of flagellin ring, but flagellin F, for example, is present in this MS ring or in S ring, and this fly G, Mn are present in the C ring. There is one additional ring that is called C ring or cytoplasmic ring, which is present inside the cytoplasm of the bacterial cell or gram-negative bacteria. Now, you can see this structure, which is a flagella, is made up of 3 things. So it is having a basal body, a hook, and a filament. You can see this hook is having this filament. So this filament is running up to this basal body. So basal body is directly linked to the filament, so that when this part rotate, so you can compare this part with the motor. So when motor rotate either clockwise or anti-clockwise, it will give some motion to the bacterial cell. Either, as you know, there are 2 kind of motions. So let me annotate this so that you can better understand. As you know, there are 2 kind of motions. I told you many times, there is run, then tumble. There is run, tumble. So actually this is run part actually. So tumble is this only when it change the direction, then run. Tumble means again changing in direction, then run. So you can see bacteria never move in a single direction, otherwise it will uh, be having fear of predators. So to avoid predators who uh, can harm the bacteria, so bacteria continuously changing its direction of motion. And you can see though this bacteria is moving in this direction, but actually it is not a single movement. It will run and then change the direction. So now you will be surprised how bacteria is able to manage this kind of motion. So this motion is with the help of this motor actually, this motor. So when this motor rotate clockwise, so this kind of motion is actually clockwise. So actually this clockwise motion is at these points. So these points are of tumble motion. So when the flagellar motor rotate clockwise, actually bacteria change its motion or its direction of movement. So this is called tumble. Tumble is always associated with the clockwise movement of this motor. Now, suppose if bacteria move this motor anti-clockwise, so then this movement of this motor anti-clockwise is responsible for this run. So the basic movements which are called run are actually the anti-clockwise or okay, so anti-clockwise movements of this particular motor. This is also called counterclockwise, anti-clockwise or counterclockwise, whatever you call it. So this will give the run motion to the bacterial cells. Now, second question you may ask me, how bacteria is able to get the energy, how it is able to move this particular motor, because this is a large supramolecular structure. So obviously bacteria need some kind of energy to rotate this motor because uh, it is made up of many proteins. As you can see, this is a heavy structure if you compare it with the overall cell, bacterial cell. So it is a prominent structure. So the movement of this particular motor needs some kind of energy. So from where this energy will come? So directly this energy will come from these proton gradients. So you can see here this is periplasmic space. So in this periplasmic space, the H positive ion concentration is maintained at a very high concentration. So it is having H positive high concentration. When these H positive will move from high concentration to low concentration, means towards the cytoplasm. So actually this will give a kind of frictional motion to this motor and help in the rotation of this particular motor, either clockwise or anti-clockwise. Fine. So it is having two proteins. This is actually, these are present on both the sides of this motor. So these are called, small protein is called motor protein A, MOTA, and large protein is called motor protein B. So you can compare this whole structure with a generator. This generator is also having two components. One is called rotor and one is called stator. So this protein, these two proteins, these are called motor proteins. These are fixed, whereas this structure is, this can rotate. So this is called rotor, these are called stator are present on both the sides. So when H positive ions will cross the plasma membrane through this channel, so it will provide a kind of channel between this stator and rotor. So it will give a kind of rotatory motion to this particular uh, flagella or this particular motor. Fine. Now we will discuss the details. So here it is also shown that this is a C ring. It is having fly M and N. Also, this is having fly F, which is a part of this MS ring. So when you see this structure, these two rings, MS ring, which are fused, and C ring, which is present in the cytoplasm, is having this structure. You can see this is very prominent motor-like structure. So it is present here actually, and when the H positive ions are passing by touching this motor and the channel between the stator and motor, they will generate a kind of torque so that this motor or this flagellar motor can rotate either clockwise or anti-clockwise. So you can see these proteins which are shown here in violet color, or you can call it pink. So these are also embedded into this particular structure. So these are stator proteins, motor A and motor B. So they will provide the channels between these motor proteins and these flagellin proteins so that H positive ions can pass between the stator and motor proteins. Each time an H positive ion will cross this membrane or pass through this channel, they will help in the rotation of this movable structure, which is called rotor. Fine. And this red colored structure which is present here is actually made up of flagellin protein G. This is made up of two type proteins, flagellin M and N. And this yellow colored structure is actually the fused rings, which is of MS ring, and that is made up of flagellin F. So you need to remember the names as well. So here it is written motor A protein and motor B protein create a channel through the which the protons can flow. This causes the flagellum to rotate. Everything is self-explanatory. You can see it is very clear from this particular molecular diagram. We will go in further details of this particular structure. Okay, so no worries. But before moving to other PDF, I would like to read few lines which are written here. So this movement releases energy. So this is a proton gradient which is present in this periplasmic space, and the protons are moving from periplasmic space into the cytoplasm through this channel. So this is a proton gradient. This movement releases energy. The movement of protons from the periplasmic space into the cytoplasm. This movement release energy that is used to rotate the flagellum. In a sense, the entry of a proton into the channel is like the entry of a person into the revolving door. You have seen in certain gardens, actually, there is a revolving door uh, in which only one person can pass at a time. So each time when a new person will come, he or she need to adjust that particular door and need to rotate that particular door so that he or she can uh, go inside the park or some garden. So the same thing is here actually. With each proton, it will give some kind of motion to this particular motor. So this is very homologous to the revolving door, as you have. I hope you have seen that somewhere. So the power of the proton generate torque, rather like a person pushing the revolving door. Indeed, the speed of flagellar rotation is directly proportional to the magnitude of the PMF. Now you can say that whatever the proton gradient here, or the proton motive force which is generating here, which is directly proportional to the concentration difference between the H positive ion concentration here and here in the cytoplasm. So the PMS, the proton motive force, which is generated as a result, these will move through this channel or through this door. They will give the rotatory motion to this particular flagella. Now, because this structure is given here and this structure is related to gram-negative bacteria, I would like to tell you this: the same structure is present in gram-positive bacteria as well. But we know that outer membrane is not found in the gram-positive bacteria. Therefore, this L and P rings are absent from generally, this L ring is not found in gram-positive bacteria. So gram-positive bacteria is having only two rings. One is present in the membrane and one is present outside the membrane, or you can say the peptidoglycan cell wall. So there is only two rings. Here you can see there are four rings. So this is the basic difference between a gram-positive bacteria and a gram-negative bacteria in terms of flagella. Gram-negative bacteria is having four rings named L, P, MS, and C ring, whereas gram-positive bacteria is having only two rings as given in your Pathfinder as well. So let me open the PDF first. So also you can see from this particular diagram, which is given in Prescott as well. Here you can see this is gram-positive bacterial flagella. So there is only two rings. One is present in the membrane, and other is present, or you can say it is. So these rings are not named like LP, MS, and C. They are simply named as. Oh, I missed. Wait a minute. So here it is given actually. So let me annotate this portion so that you can read this out. So it is given that there is four rings: LP, MS, and C. are connected to a central P for peptidoglycan, L for lipopolysaccharide layer. Later it was found that MS rings are now called the C ring for cytoplasmic is present inside the cell. The C ring consists of three proteins, G, M, and N. We have discussed this. Typical gram-positive bacteria have only two rings. In this, it is called inner ring, which is connected to the plasma membrane, and there is an outer ring, which is probably attached to the peptidoglycan cell wall. So they are named as outer ring and inner ring. The inner ring is connected to the plasma membrane, and outer ring is connected to probably connected to the peptidoglycan, which is actually not connected, as you can see from this particular diagram. So there are differences in the flagellar structure, as we have seen in gram-positive and gram-negative bacteria. For reference, you can find this particular diagram in your Prescott, which is given page number. I will tell you, no problem. 75. Okay. So let's discuss flagella in detail so that we can also cover pilus and Fimbriae portion also. So bacterial flagella are slender, rigid structure about 20 nanometer across up and 20 micrometer long. It is a long structure because you can see here it is 20 micrometer long. Micron is 10 to power minus 6 and nanometer is actually 10 to power minus 9. So you can see the length of flagella is more because it is in micron, whereas the structure which is embedded in the cell is only 20 nanometer. Flagella are so thin that they cannot be observed directly with the bright field microscopy or some light microscope tool. As we have discussed in our previous classes also, we need electron microscope to visualize flagella in a better way. Then with how they are present, depending upon their position over a bacterial cell, they can be monotrichous. Trichous mean here in Latin, in Greek word, in Greek terms or in Latin. So heaven flagella, it is located at one end. Polar flagellum भी बोलते हैं, and then there is amphitrichus. Amphi means when the flagella is present both the sides of bacterial cell. Lophotrichus, when lopho means a bunch or tuft, when a cluster of flagella is present over one end. So this is Lophotrichus, which is shown here. Uh, they are having only one flagella, therefore they're called monotrichous or polar flagella. If one flagella is present over this end also, then they are called amphitrichous. So here it is given Lophotrichus, it is present in spirillum. Then these are peritrichous actually. So peritrichous means that when the bacterial flagella are present all over the body of the bacterial cell. Peri means around. Uh, so in Pseudomonas, mostly there are monotrichous polar flagella are found. Lophotrichous are found in spirillum, and peritrichous is found in Proteus vulgaris. So examples are also given in this text. Now, they are found both in bacteria and archaea, so there is no problem. Okay, in this diagram, they have shown that along with flagella, so flagella are more prominent structures. You can see they are also thread-like structures, but they are thick in comparison to other structures which are also found in bacteria, either Fimbriae or Pilus. So they are having different roles. For example, these Fimbriae are helping the bacterial cell in attachment to the substrate or the host cells, and pilus is having another role. Pilus, for example, sex pilus is responsible for the movement or the transduction, means movement of plasmid from one bacterial cell to the another bacteria through this pilus. So transduction transformation is a function of pilus, whereas Fimbriae are helping the bacteria to attach to the surfaces either in the environment or to the host. It is given here peritricus, peri means around. Now, basic question. This flagella is made up of three parts. Filament, there is a hook-like structure. Filament is most obvious portion because it is a long structure which is extended from the cell surface up to the tip. Basal body is embedded into the cell and a curved segment which is present between the filament and basal body. This is called hook. Made up of protein flagellin, I have already told you this is having molecular weight ranging from 30 kilodalton to 60 kilodalton and always depend upon the species of bacteria. Okay, now there is one more important thing is in some species there is some capping proteins and some species they are absent. Some bacteria having a sheath-like structure which is surrounding their flagella. For example, Vibrio cholerae, which is a causative agent of cholera, having this lipopolysaccharide sheath over their flagella. Okay, so this portion actually we have discussed about gram-positive and gram-negative bacteria. They are having four rings. Gram-positive bacteria is having two ring only. Inner ring connected to the plasma membrane, outer ring probably attached to the peptidoglycan. Actually, the Pathfinder is directly copied from this particular book. The same line is actually written in the Pathfinder as well. Okay, now this flagellin protein, because there are different type of flagellin, we have seen flagellin F, G, and N, and also there are different proteins. As you can see from this particular diagram, I will show you one more diagram. Uh, so there are two important things you need to note. First of all, so this is the filament, this is hook, and there is a basal body which is embedded in the cell. There are certain different type of proteins, flagellin B1, which is a major flagellin, flagellin B2, and there are other which is not shown in this diagram. So I have another. So flagellin B1, B2, B3, and flagellin A. All they can be distinguished by this color. So the different shades of blue, flagellin B1, B2, B3, and flagellin A, they are present in the four different type of flagellin proteins are found in filament of flagella. Then this flagellin A is mainly found in the hook structure. Also, there are some other type of flagellin protein, for example, flagellin H, J, flagellin I. So they are also found, and either they are making the stator or the rotor portion of this particular flagella. At the same time, this ATP is getting converted into eight. And so this is a kind of ATP is flagellin I, which is present in this the structure of flagella. This flagellin is a kind of ATPase. It will convert ATP into ADP and will give some kind of energy to so that helping the flagella to rotate. Okay, so the structure here is actually for archaeal flagella. Up to here, we have discussed bacterial flagella. So there are some or a little few differences between the structural organization of bacterial flagella and archaeal flagella. So the archaea is having very simple structure of their flagella, whereas bacterial flagella are having more pronounced ring-like structures, as we have seen from this particular diagrams. So gram-negative bacteria and gram-positive bacteria and their flagella are quite different in terms of molecular structure. And if you compare them with archaeal flagella, so archaeal flagella is completely different. It is the simplest flagella found in bacterial cells. So there is a query. Okay, so flagellin protein clear diagram for once. Okay. So, Trishla, we have compared two different types of flagella in gram-positive bacteria and gram-negative bacteria. So, both are bacterial flagella. Then we have compared this eubacterial flagella with archeal flagella. So there are differences in the structure and organization of flagellin proteins, as you can see in this, because there are pronounced rings, ring-like structures, and in each ring there are different proteins. But here, this is a simple structure. So this type of flagella is actually found in archaea. So in the filament, you can see there are different proteins: flagellin B1, B2, B3, and flagellin A. Flagellin A is mainly making the hook of archeal flagella, and there are certain different flagellin proteins called H, J, and I, which are not present in bacterial flagella, but they are present in archeal flagella. Flagellin I is having some role. It is found to be ATPase, which is hydrolyzing the ATP. There are certain other flagellin proteins. We do not know the names of these proteins, so there is a question mark. They are present, but they are not named yet. Okay, so there are different type of flagellate proteins which are together making the archeal flagella. Then if you compare this flagella with the bacterial ones, so see here again there is one more structure. This I showed you in the, let me zoom out this. So it is having flagellin M and G in the C ring and flagellin F in the MS ring, which are the major flagellin proteins found in the bacterial flagella. After the class, when you compare them, you can easily differentiate them because they vary not in their structural organization, but also in terms of their protein organizations. Okay. So this is flagellin A protein, which is in archeal flagella. This is the minor portion only. Uh, Flagellin B3 is present in this hook, and the major flagellin which are found in archaea are B1 and B2. Priyanshi is having a question: do archeal flagella also have rotatory motion? Priyanshi, yes, because bacterial flagella, whether it is archeal flagella or means this. Okay, so we also compare this flagella with eukaryotic flagella as it is given in your Pathfinder as well. Uh, so let me quickly cover this portion so that. Okay, so this table you can see it is given in your Pathfinder page number 432. Bacterial flagella, whether it is gram means the bacterial flagella, eubacterial flagella, or archeal flagella, it is rigid structure. It is a rigid structure because so many flagellin proteins are present. Eukaryotic flagella is flexible in terms of, because it is having mainly tubulin proteins. Okay, so when it is flexible, it will have some other movement possible. But if it is rigid, the flagella can only rotate, means it will have mostly perform rotatory movement, and it is not surrounded with plasma membrane, whereas the eukaryotic flagella is flexible, made up of tubulin protein, tubulin alpha and beta proteins, perform whiplash-like movement. So in terms of movement, eukaryotic flagella is completely different from bacterial flagella, and it is so because it is flexible in nature, therefore it can have a whiplash-like movement, whereas bacterial flagella are mostly rotatory in nature. It is surrounded with the plasma membrane, but it is not surrounded with plasma membrane. But as we have discussed, in some species of bacteria, it is covered with either proteins or some kind of sheath, or either LPS layer, lipopolysaccharide layer. So these are some differences between the this we have already discussed monotrichous, amphitrichous, lophotrichous, and peritrichous flagella. Depending upon the position of flagella in the bacterial cell, they are named like this. And atrichous is also possible if the bacteria do not having the flagella at all. So this is also possible. Atrichous is also. So now we are interested in bacterial flagella, or you can say the eubacterial flagella. Why so? Because uh, archeal flagella is very simple organization, and the second thing is many proteins which are present or found in the archeal flagella, we do not know about the names of those particular flagellant proteins. So therefore, they are shown with the help of one question mark here. Oh, sorry, I have. Okay, so this diagram is also important. As you can see here, the two type of motions are shown here. So if you put this, if you see this is a kind of anti-clockwise motion. So when this flagella is moving anti-clockwise, it will forward, it will show the forward run. Whereas if this flagella will rotate clockwise, okay, so it will show the tumble movement. In order to move the flagella to run or to move it forward, if it is having more than one flagella, all should move in this anti-clockwise direction so that the flagella will move in it will show kind of a run. But if a bacterial cell will have more than one flagella and all will rotate clockwise, you can see here, then this cell will show the change in direction, means it will tumble around. So now you are able to understand how the bacterial cell is able to manage the movement, either if it want to run, or if it want to change the direction, or it want to tumble around. Okay, so if it will rotate counterclockwise, it will run. If it will rotate clockwise, the cell will tumble around, or ultimately the direction of the bacterial cell movement will change. There is one important topic that is called chemotaxis. Chemotaxis is the term when bacterial cells start moving in the gradient of the some chemical stimulus or nutrient. So four methods of movement has been observed in bacteria. Uh, okay, they are swimming movement. If you are not able to see, please see here. Swimming movement with the help of flagella, corkscrew movement as seen in the spirochetes, twitching motility, it is associated with pilus. So now remember, pilus will give the two advantages to the cell. Either it will help in the movement. This is linked to the twitching motility of the bacterial cells. Also, this pilus is responsible for the transfer of DNA from one cell to other cell, means the transformation kind of. And the fourth movement is gliding motility. So four basic movement has been seen in bacteria. Okay, so first of all, I will show you these four basic movements so that you are able to better understand this, what we have discussed. Not given in this. So here they are shown actually. So, for example, this is called spirochete flagella. Numerous flagella arise from each end of the spirochete. Uh, there are large number of flagella which are arising from the, as we know, spirochete is a bacteria itself. Okay, so it is having some spiral shape, therefore it is called so. These intertwine to form an axial fibril. Axial fibril wind around the cell, usually overlapping the middle. Treponema species showing the three flagella arising from the tip of the cell. You can see here there are three flagella which are arising from the tip of the cell, and they are together giving this kind of movement. These are not very important, but at least you need to. Okay, there is a query. Pilus help in movement of some bacteria. So those bacteria lack filament or is the additional function of bacterial cell. Okay, quite interesting. Uh, Type 4 pilus, which we have discussed. Okay, so it will both way possible. Either they do not have a flagella at all, they are atrichous, or it can also give some kind of advantage to the flagella in the movement. But the okay, so as we have discussed the four type of movements, either spirochete, twitching, gliding, and all these. So somewhere they will involving this pilus as well. For example, here, so if the pilus is present, it will give some different kind of motion to that particular bacterial cell. So it is given here, this type 4 pilus, which is present in some bacteria, it will help the bacteria in this movement. This is called twitching and gliding motility. Occur when cells are on a solid surface. Both type of motility can involve the type 4 pilus. Okay. If you want to read more about this, these different type of movements which are seen in bacteria, you can refer this PDF. So it is also given here. There are some species like Flavobacterium species, Mycoplasma species, which are distinctly considered in detail in the upcoming chapters. Type 4 pilus is present at one or both poles of some of the bacteria and are involved in twitching motility and in gliding motility. In some bacteria, twitching motility is a characteristic short, intermittent, jerky motions of up to several micrometers in length and is normally seen on very moist surfaces or some. So these are solid surfaces, but they're moist in nature. It occurs only when cells are in contact with each other. Isolated cells rarely move by this mechanism. So suppose there are large number of cells which are making a bunch-like structure. So only in these circumstances, this twitching motility has been seen, and this is purely due to the presence of this type 4 pilus. Okay. So what you need to remember, though this is not very important section what we are referring, but at least you should know this twitching motility or this gliding motility is due to the presence of type 4 pilus. It is only seen when there are moist solid surfaces and the cells are present in contact with one another. When the cells are present isolated, these type of movements are generally not seen. You need to remember this. There is one more query. Okay, there is no query. So we should move. So the important things actually I have covered. I need to tell you actually there are different type of motions which can also have been seen in the bacteria. These are called twitching motility, gliding motility, spirochete flagellum or the spirochete movement, and so on. After this, actually, we should discuss chemotaxis in a small detail. So this chemotaxis is actually the movement of bacteria, so movement of cells towards some chemical attractant or away from this particular chemical species. This is repellent. So this is called chemotaxis. Both way possible, either towards the chemical species or away from the chemical species or danger. So this is a kind of gradient based movement, means the bacterial cell will move in a concentration gradient of this chemical species. And this is shown here. The bacteria generally move like this: run, tumble, and so on. So first of all, the first movement is very random, so you can name them as well. So suppose bacteria is present in such an environment when there is no chemical attractant at all. So this can be random. So bacteria can move, can go away, and can remain at the same place because there is no attractant. Or suppose there is some chemical species present at this point. So suppose the concentration is very, very much here. What will happen? A bacteria will start moving from this side and will move to the other end according to the concentration gradient. So as you can see, there is a green shade shown here. So green shade is actually the concentration gradient of that particular species. So the bacterial cell will show the movement in an attractant gradient. Tumbling frequently is reduced when the bacteria is moving in the some kind of gradient. Therefore, runs will become longer. You can compare these runs. These are shorter actually. You can compare these runs are longer. So actually, this kind of biased movement also has been seen in bacteria. This is random movement when there is no chemical gradient, and if there is some nutrient present over this end, bacteria will cells will less tumble and will show long runs, so that bacteria can bacterial cell can have one directional movement. Okay, so that it can reach to this particular site where nutrients are present in high concentration. Have a query, or are you able to understand this point? This is important. If you have certain query, please ask me at this point. Otherwise, so we have discussed these proteins which are present in gram-negative bacteria. As you can see, the most complex flagella, which is present in gram-negative bacteria itself, because it is having 4 rings. In gram-positive bacteria, it is having only 2 rings, so not that much complex. So this is the most complex flagella found in gram-negative bacteria itself. Now you can see there are different type of proteins which are which we have discussed actually. Flagellin G, M, N. These are making the C ring. This is called the motor switch, also called switch complex. The switch complex or the C ring is attached to MS ring. MS ring is made up of fly F, which is not given here, but as we have seen from the Prescott. So this is having fly F proteins, flagellin F protein. Then there are 2 additional rings, L ring in the lipopolysaccharide layer and P ring in the peptidoglycan layer. And finally, these 2 proteins, protein A and motor protein B, are making the stator. And between them, there is a little gap. So this will provide a channel so that the H positive ions can move from the periplasmic space into the cytoplasm. There is a question: chemotaxis moves towards the nutrient to consume for their energy, right? Does they consume from the chemical producing bacteria by attaching with them? Quite confusing. You are not clear what you want to ask, Trishla. Please be.
हेलो सर।
हाँ हाँ बोलो.
सर मेरा क्वेश्चन ये है, जो कीमोटैक्सेस हैं वो मूव कर रहे हैं न्यूट्रिएंट्स की तरफ।
And they are not involved in the consumption of nutrients. The bacteria is having some other ways to consume the nutrient. Okay. So this we have discussed switch protein complex, which is actually made up of 3 different flagellin proteins. Again, I'm telling you, this switch complex or motor switch is actually called the C ring. It is made up of 3 protein G, M, N in the sequence. And why this is called switch complex? Because this switch complex, as it can switch the direction of the flagellar motor. either clockwise or anti-clockwise. So therefore, the C-ring is the culprit whether the bacterial flagella will move clockwise or anti-clockwise. So it will decide. Then bacterial flagella motion is rotatory in nature. As I told you, the exact mechanism that derives the basal body rotation, it is still not very clear, but we have some assumptions. As one assumption is, uh, with the help of these motor proteins, it will give some kind of motion to this particular motor. And the second assumption is when the protons are moving from the periplasmic space into the cytoplasm, they will help in the rotation of this particular motor. They are assumptions only. This is not very clear how the bacteria is able to rotate this its basal body. The necessary free energy is derived from the proton gradient that exists across the plasma membrane. ATP is not directly required for the flagellar motion, but it is assured that bacteria like unlike archaea, as we have seen in archaea, ATP is directly consumed by flagellin I protein. But here in the bacteria, the ATP is not directly consumed, but the H positive ion gradient is actually used to rotate this particular flagella. Membrane proteins, motor A and motor B create a proton channel that drives the rotation of the flagella. We have discussed this rotation of flagella can be clockwise, counterclockwise. Now you need to underline this. If it will rotate clockwise, it will tumble. If it will rotate counterclockwise or anticlockwise, the bacterial will show the forward motion that is called run. This table we have discussed. So the difference in the bacterial and eukaryotic flagella are in the, first of all, in the structure. It is rigid, it is flexible. Because it is flexible, it can show the whiplash-like movement. Um, okay, but it is having this rotatory motion. Also, their makeup is different. Flagellin proteins, here they are having tubulin proteins. So this is all what we have given, what is given in your book about this flagella or the flagellar movement. Then bacteria is also having some other structures or surface appendages which are also required for the bacterial cell. So let us discuss and quickly finish this portion at least. So pilus. Pilus is used to describe the thin hair-like appendage on the surface of bacteria. You know much about them because pilus is actually we have discussed two type of pilus. One is called type 4 pilus, which is responsible for gliding and twitching motility in bacteria when they are present along with other bacterial cells, not in isolation. And the Fimbriae, which are responsible for attachment of these bacteria to the substrate or to the host cells. So here, like appendage on the surface of bacteria, pilus for single, and if it is simply, it is regarding when they are present in plural form. Gram-negative bacteria, almost exclusively on gram-negative bacteria. Gram-positive bacteria generally do not have pilus. Only a few gram-positive bacteria are found to have this. Exceptions are always there. So, for example, Corynebacterium renale is having pilus, which is a gram-positive bacteria. But mostly, gram-positive bacteria do not have pilus. It is mostly found in gram-negative bacteria only. Proteins that form the pilus are referred to as pillin proteins. Now the proteins also change. In flagella, flagellin proteins are found. In pilus, the pillin proteins are abundant. Pilus are more rigid in appearance than flagella. So we have discussed the flagella, and we know flagella are rigid structures, but pilus are even more rigid. It plays an important role in process of conjugation. Conjugation is therefore they're called sex pilus. It is given here. It is also function as a receptor for donor specific. Donor is called male. So the bacteria which is giving the plasmid or the DNA is called donor, and there is an acceptor bacteria. So there is a tube-like structure which is formed between a donor and a acceptor, so that this DNA can be transferred through the process of conjugation. Now there is second structure, Fimbriae. Fimbriae filamentous cell appendages present in both gram-positive and gram-negative bacteria. As the pilus is found mostly in gram-negative bacteria, pilus are found only in gram-negative bacteria. Fimbriae are abundant in both gram-positive as well as in gram-negative bacteria. They're composed of helically arranged protein subunits, and their main function is to mediate the cell attachment to the surfaces, either to the host cell or to the some kind of attachment to the, so that the bacterial cell can adhere itself to some substrate. It can also add the poles of bacterial cells, so can evenly distribute over the entire surface of the cell, and depends upon the species whether they are present in the at a single place or they are abundant all over the cell in the bacteria. Their number also vary from one cell to another species, so few to several hundred per cell. Then there is a table which is giving the differences between pilus and fimbriae. So mostly their number vary. Pilus are mostly found in gram-negative bacteria, whereas they are found in both gram-positive and gram-negative bacteria, as we have discussed, required for conjugation, but they are required for cell attachment to the surfaces. Their number is less. They are found generally 1 to 10 per cell, but fimbriae are more because attachment is directly proportional to the number of fimbriae present over the bacterial cell. So their number can be up to 1,000 or so per cell, and they are short in length when compared to pilus. So there are certain differences between the pilus and fimbriae. Okay, so the next topic which is given in your Pathfinder is endospore. Why it is called endospore? Because not all the bacterial species making endospores, first of all. Second, it is called endospore because these are formed inside the cell. Endo means inside. Spore means kind of structure which is very resistant to the environmental stresses, for example, UV radiation, heat, cold, and so on. For example, some bacteria are reported which make endospores when they are having some kind of stress, for example, when there is nutrient deficiency. So, because these spores are having less metabolic activity, and therefore it is given in this table, they are very high heat resistance, radiation resistant, for example UV resistance. Their water content is very low, only having generally the water content of a living cell is, or the vegetative cell is 70 to 80 or up to 90% in bacteria. But in endospore, if they're making endospores, so endospores are having only 10 to 25% water in the core. pH is also less. Generally, the vegetative cell having around neutral pH, but they are having 5.5 or 6 or so. What is why they are very resistant to the heat and radiations and other kind of stresses? Because of two reasons. One is dipicolonic acid. This dipicolonic acid and calcium content is very high. So these endospores are having high calcium content, and this particular substance, dipicolinic acid, is present only in the endospores, not in the vegetative cells. So these two additional components which are found in these endospores are responsible for all the stress resistance, for example, heat and radiation resistance inside these endospores. And you can see there is low enzymatic activity, and mRNA is low or absent because they are very less metabolically active. So their metabolic activity is very, very less. It will decrease. So therefore, they do not require energy or nutrients in very high concentration. So now the question is, what species of bacteria are making the endospores? Mainly Bacillus and Clostridium. So these two species are most prominently found to make endospores. Exceptionally heat resistant dehydrated structures which are capable of surviving for long periods of time in unfavorable conditions of environment. These are very dormant structures. Dormant in the sense they are resistant to these stresses, called endospores, since they develop within the cells. Within the cell, therefore called endospores. Ordinarily resistant to the environmental stresses, for example, heat, UV radiation, gamma radiation, chemical disinfectants, and even desiccation, because they are already having less water content, therefore they are very resistant to desiccation. So that's all which is given in your book. So there is a structure which is given in Prescott. Depending upon the position of the endospore, they can be central, subterminal when they present towards one end of the bacterial cell, terminal very present at the end of or the pole of one's bacterial cell, or they are swollen sporangium, means the spore bearing part is actually having some kind of swollen appearance. So depending upon their position, the endospore can be categorized in either, there are four types. Okay. So we will discuss about endospores in more detail in our next classes. But a quick, so that you are able to understand something about endospores. So there is a diagram. As we have discussed, they can be stained with Schaeffer-Fulton dye, which is a combination of malachite green and saffron in counter stain. So therefore, it is giving two kind of colors. One is green color, so endospore is green in color, whereas the outer portion is pink or red, which is stained with counter stain saffronin. Okay, so this is a pars. This is a general endospore. So bacterial endospore. It is isolated from Bacillus subtilis, as Bacillus and Clostridium can make the endospores. Now, their structure is very important because they are formed inside the cell, bacterial cell, they're called endospores. Depending upon their position, this is a kind of subterminal endospore. Now, the endospore is made up of an outer coat. So, this is outer coat, as you can see from this and this diagram. Wait a minute. So this is a kind of endospore. The parts or the components of endospore are outer coat. There is an inner coat. Then there is cortex region. Cortex is this heavy blue colored region here. Cortex. There is a core, and because the endospore is also having this nucleoid region, so where this nucleic acid or the DNA is present, and there is core wall, which is differentiating or which is having this core portion. So this complete is core. In the core only this nucleoid region is or the DNA containing portion is present. So there's one more structure actually. So I can show you. Okay, so the question is how these endospores can be formed within bacterial cells. So suppose a bacterial cell is dividing by normal cell division, it will make an equal sized cells. But what will happen? An axial filament formation will happen. So axial filament formation within a cell. So we are taking only one cell this time. And when the cell will divide, there is an unequal division. So it will make a spore mother cell, and this will engulf the small cell. Okay, so when there is an unequal division, so this is a small cell, this is a large cell, so it will lead to the development of a four spore. So this will lead to the development of this spore. Then this big cell will engulf this small cell. So it will become endospore, and this will become the spore mother cell. So for example, see this engulfment of this four spores. So this cell is uh, being engulfed by this particular cell, and when this will engulf this particular small cell, this will become inside this, therefore develop endospore. So endospore will be further differentiated. It will develop cortex region, exosporium coat. So this exosporium will coming all over the endospore, called exosporium. Exosporium and this endospore is having a membrane in between them. This is called spore coat, and spore coat is outer coat and inner coat, and finally lead to the development of this endospore. So this endospore is having exosporium. This is the spore coat. This is the cortex region. And finally, this is the core where the nucleoid region is present. So now you can see this particular diagram. Now it is easy to understand this diagrams. So this is the core. This core will have this nucleoid region where the DNA is present. Cortex, which is the outer region. Then it will have a inner and outer coat, and there is a core wall uh, which will limit this core region. Fine. Actually, it is not shown here outside this cortex and spore coat. So suppose this is a spore coat, inner coat and outer coat. There is exosporium is also present outside this endospore when this is a free endospore. So it will also have one extra layer that is called exosporium. So these are all the components of this particular endospore, and this is the process of sporulation, how the two cells are formed. One forespore will be engulfed by this big cell, and this will lead to the development of endospore inside the cell. And when the conditions are favorable, the endospore will germinate to give the endospore germination. During the germination, the spore will emerge out of this cell. It is also given in your Pathfinder, so let me underline what you need to remember from this discussion. So endospores are very heat resistant. You should know this. They're extensively heat resistant. Why they are heat resistant? Because they are having a dipicolinic acid and the calcium concentration high. So it will give some stress resistance to these endospores. Scaffold filtering dye is used to stain the endospores, and the scaffold filtering dye is having this primary stain is malachite green, and the secondary stain or the counter stain is saffronin. You need to heat so that malachite green will enter the endospore. After this, you need to remember. Okay, so all the structures are given. Exosporium, it is a thin, delicate protein covering over the endospore. Spore coat is made up of two layers. Actually, it is several layers, but there are two major layers: inner coat and outer coat. Lies beneath the exosporium, composed of several layers. Cortex region present beneath the spore coat, and it is made up of peptidoglycan. So you need to remember the composition actually. So, for example, exosporium is a protein covering. Then cortex is made up of peptidoglycan cell wall material, which is present in the bacteria as well. Spore cell wall or the core wall, which is limiting the core of endospore, present beneath the cortex, surrounds the protoplast or the spore coat, spore core actually. So the spore core, so the core wall, normal cell structure, which is having ribosomes, nucleoid region, and so on, and but it is metabolically inactive. So this is the major characteristic of a spore. It is a dormant structure formed under unfavorable environment conditions. Therefore, it will be metabolically inactive. Large amounts of dipicolinic acid complex with the calcium ions. Therefore, it will give some kind of stress resistance to the. This has been asked actually. So this is a question from one of the computer examinations. Why this is so much stress resistant? Because it will have large amount of dipicolinic acid, which is making complex with calcium ions. Therefore, contribute the endospore heat resistance as well as some other UV resistance and so on. Question: chlorine treatment can use to kill the endospore. Though these are very resistant to all kind of chemical treatments as well as from the UV radiations are not able to kill the endospores in sometimes. Chlorine treatment. Okay. I'm not sure actually. I will see whether chlorine can be used to kill the endospores or. Okay, so after class I will confirm this. I'm not very sure whether chlorine can kill the endospores or. Actually, there are some chemical treatment which can be used to remove the endospores as well. But they are resistant to most of the stresses like UV and most chemicals. Okay, so the main reason why they are so much stress resistant is only because they are having dipicolinic acid and calcium ions. Now, the formation of endospore we have discussed from the Prescott. There is vegetative cell when it is facing starvation, and there is nutrient deprivation. Deprivation. So, what will happen? Sporulation will begin. The first morphological changes in the transformation of the two identical chromosomes, the parent chromosome and a newly replicated daughter chromosome, into an axial filament of DNA. Septum then divides the cell unequally, and the smaller prespore cell or forespore cell is engulfed by the spore mother cell. So spore mother cell is large and prespore cell is small in size. In this cell, within a cell like structure will form. We need to remember this endospore is a cell within a cell structure. So plasma membrane separate the cytoplasm of one cell from the cytoplasm of the another cell, leaving some space in between this. The space is the spore wall material will be deposited. So it will make the spore wall. So it is given here, cortex region, you can call it cortex or. Okay, so in this region, the spore wall material will be deposited. It will make several layers, exosporium, then spore coat, and then this cortex region. This core will actually the cell is present in this core. This core is this four spore cell which is being engulfed by this big cell, because it will have nucleoid region as well as the small ribosome, 70s ribosomes. So for proper understanding of the formation of endospore, you can refer this particular diagram. It is given in the steps here: cortex formation, the coat synthesis, and then completion of the coat synthesis, and heat resistance formation, lysis of sporangium, and the liberation of free spores, so that they can germinate in favorable conditions. Okay, so at last there are differences what we have covered in our previous classes as well. So what are the differences between a gram positive and gram negative bacteria? There are gram negative. Gram negative is having flagellar differences we have covered today. Along with flagellar differences, they are having thin cell wall. So they are 2 to 7 nanometer thick cell wall only. Whereas the gram positive bacteria is having peptidoglycan cell wall, which is very prominent, 20 to 80 nanometer thick. Teichoic acid is only found in gram positive bacteria. Periplasmic space is prominent in gram negative bacteria because it is the space present between the outer membrane and plasma membrane. Outer membrane only present in gram-negative bacteria. Motility. Gram-negative bacteria is mostly motile. It can be non-motile, but actually, gram-positive bacteria is mostly non-motile in nature. Okay, so because the flagella or other type of structures, for example, pilus, type 4 pilus are mostly prominent in gram-negative bacteria. So they are motile. They are mostly non-motile. Appendages. इनमें सभी होते हैं तो pilus and Fimbriae they are found in gram-negative bacteria, but they will not have the pilus mostly, so they will lack some of the appendages. For example, pilus lack pilus. Four rings in their flagellar basal body, but there is only two rings. This we have discussed today only. Toxin is produced, so gram-negative bacteria is having lipopolysaccharide layer, therefore they will produce endotoxins, whereas they will produce only exotoxins. Gram-negative bacteria can also produce exotoxins, but endotoxin is common to gram-negative bacteria only. Endospores, negative bacteria cannot form endospores, but some genera of gram-positive bacteria like Bacillus and Clostridium, they are generally making endospores. Gram-negative bacteria will not retain the crystal violet dye, therefore they does not retain the purple color, and therefore when they are counterstained with safranin, so they will give some pink color or some red color, whereas the gram-positive bacteria violet in nature because they will be stained with crystal violet, so they will see in like purple color under the microscope. And we have discussed their differences in their membrane. So outer membrane is having this structure. These are called porins through which low molecular weight compound can cross this outer membrane in gram-negative bacteria. Whereas gram-positive bacteria is having a thick cell wall, 20 to 80 nanometer thick. This cell wall is very thin, and only 2 to 7 nanometer thick in gram-negative bacteria. This is a cell wall of gram-positive bacteria. It is very thick, 20 to 70 nanometer. So that's all we have covered bacterial structures, surface appendages, endospores in this class. And the next topic is bacterial genome. Genome can be of two types: bacterial chromosome, which is present in nuclear region, and mostly the most of bacteria is having plasmids. Different type of plasmids are present in bacteria, found in bacteria. So depending upon plasmid presence, they can be categorized. So we will discuss this genome section in next class. That's all for today. If you are having queries, please ask. Otherwise.